All right, today we're cracking open a case file on a whole family of microscopic criminals. We're going to be profiling some of history's most notorious pathogens, and we're starting with the big one, the absolute mastermind behind the Black Death.
Just think about that question for a second. It's one of the most terrifying questions in all of human history. We're talking about a pandemic so devastating it literally reshaped societies, a killer so effective they just called it the Great Mortality. So, what kind of monster could possibly be responsible for something like that?
Well, here it is. Meet the culprit. This little thing is Yersinia pestis, the plague bacillus. It looks pretty unassuming, right? Just a tiny rod-shaped bacterium. But under the right, or, wrong conditions, it becomes one of the most brutally efficient killers this planet has ever seen. So, let's start our investigation with the prime suspect. And you might think, oh, the plague, that's ancient history. But let me tell you, this is far from a cold case. This organism is still out there, living in pockets all around the world. A constant, lingering threat.
So what exactly are we looking at here? Well, first, it's a gram-negative bacterium that just tells scientists a bit about its cell structure. It's also a coccobacillus, which basically means it's shaped somewhere between a little sphere and a rod. But here's the key thing. Its natural home, its main hideout, isn't in people. It's in rodents. We're just the unfortunate victims who get in the way.
Now, to really understand how this tiny bacterium caused so much chaos, we've got to dig into its MO, its modus operandi. How does it find its victims, and what does it do that's so incredibly devastating?
The whole operation relies on this classic three-step cycle. It's simple, but it's brilliant. Step 1, the bacteria hangs out in rodents. Step 2, a flea comes along, bites an infected rodent, and becomes the getaway driver, the vector. And step 3, that infected flea then jumps onto a human, bites them, and injects the pathogen right into a brand new host. It's an ancient and, frankly, terrifyingly effective delivery system.
Okay, so once it's inside a person, the plague can show up in 3 different ways, 3 distinct phases. The most famous, of course, is bubonic plague. That's from the flea bite, and it leads to these horribly swollen lymph nodes called buboes. Now, if the infection gets into the lungs from there, it becomes pneumonic plague. And this is the most dangerous form, because it can spread directly from person to person through coughing. And finally, if it gets into the bloodstream, it's called septicemic plague. This is what causes the widespread internal bleeding that gave the Black Death its chilling name.
Now check this out. Here's a really cool clue for the microbial investigators. When you put a stain on Y. pestis and look at it under a microscope, it shows what's called bipolar staining. The ends of the bacterium soak up way more dye than the middle part, and what you get is this classic safety pin shape, a key piece of visual evidence that screams, "You've found your culprit."
And here's how they gather even more evidence back at the lab. You take a sample and grow it on a blood agar plate. Yersinia pestis forms these distinct, small, almost see-through colonies. This is basically the microbiological version of collecting fingerprints at a crime scene. It's how scientists can be 100% sure of the suspect's identity.
But here's the thing. Yersinia pestis isn't a lone wolf. Nope, it's part of a larger criminal family, the whole Yersinia genus, and it has a couple of other relatives that cause their own special kinds of trouble.
So meet Yersinia enterocollitica. Its MO is totally different. Instead of using a flea, its crime scene is usually contaminated food. Think undercooked pork, for example. And its main offense isn't the world-ending plague, but a really nasty intestinal illness. So, a different method, but definitely from the same criminal family tree.
And then there's the third member of the family, Yersinia pseudotuberculosis. As its name kind of suggests, it can cause a disease that looks a bit like tuberculosis, forming these little white spots on organs. It also gets to people through contaminated food or water, and it can cause anything from mild stomach issues to a severe typhoid-like sickness that can actually be fatal.
Okay, let's widen our investigation a little bit to look at some known associates. These are other germs that are also spread by animals. And for a while, scientists thought they were all part of the same criminal gang.
And this is a really fascinating bit of microbial forensics. See, back in the day, scientists had all these organisms lumped together under one name, Pasteurella. But as our genetic techniques got way better, they realized they were dealing with totally distinct criminals. It was like a detective using DNA to discover that members of a supposed crime family weren't actually related at all.
First up is an associate that many of us literally live with every single day, Pasteurella multicida. This little guy is super common in the mouths of perfectly healthy cats and dogs. The danger comes from a bite or a deep scratch, which can push the bacteria deep into your tissues and lead to some really serious skin or even bone infections.
And that brings us to a very close associate, Francisella tularensis. Now this one really shows the connection back to our main suspect. It causes a disease called tularemia, which is often described as looking a whole lot like bubonic plague. It's also passed on from animals, usually through things like tick bites or by handling infected wildlife. A real copycat.
So, we've got our lineup of suspects and their known associates. The big question now is, how do we fight back? How does modern science bring these microscopic criminals to justice?
Well, the first step is identification, and let me tell you, it is a high-stakes game. Handling a suspect like Yersinia pestis is so dangerous it has to be done in a special Class 3 containment lab. You confirm the diagnosis by actually finding the bacteria in samples, like fluid from a bubo or in the blood, using all our modern forensic tools, like growing cultures, doing antibody tests, and of course, PCR.
Once you've got a positive ID, the response has to be fast. Our go-to weapons are heavy-duty antibiotics like streptomycin and tetracycline. But here's a critical detail, a real curveball. Yersinia pestis is completely resistant to penicillin. It just doesn't work. Knowing that one fact is absolutely vital for saving lives. And of course, patients have to be isolated to stop the crime spree, especially if they have that highly contagious pneumonic form.
But in the end, the best way to bring these guys to justice is to stop the crime before it even starts. And that means strong public health defenses. Keeping an eye on animal populations, controlling rats and fleas, and making sure people have good sanitation. For anyone who gets exposed, preventative antibiotics can be a lifesaver, and yeah, in some high-risk parts of the world, there are even vaccines as another line of defense.
Which really brings us to our final thought. It's so easy to think of the Black Death as something from the history books, right? But the culprit, Yersinia pestis, and its whole criminal family, they're still out there. They're a constant, quiet reminder of just how delicate the balance is between us and the microbial world. So the real question isn't whether these threats will pop up again, but how ready we'll be when they do. What have we really truly learned from history's deadliest suspect?